Hi, Lydia. Can you hear me okay?
Yes, all working fine.
Brilliant. My camera doesn't seem to want to work, which is quite interesting.
We can see you.
Yeah, it's not the it's my
Oh,
emergency camera
okay.
rather than the main one, but let's see what we can do about that. What this also means is that my slides won't appear on the screen I want them to.
If you want me to share your slides, you can just send them
Oh
over.
no, it's more that they won't. I need to put it back onto presentation mode, and I want the main screen. I want the slides to be on the screen, so I'm looking at people.
Okay.
Slideshow automatic. Maybe it's that's going to be the worst of. Okay, that may just. If I share that, is that working there, Lydia?
Yeah, we can see that.
And you're seeing just the slides?
Yeah, just the slides.
Excellent, 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 excellent. And do we have Honghan here?
Yeah, I'm here.
Perfect. Morning, Honghan. Excellent. So I think it's me first, and then we hand over to you. And brilliant. Okay. I'm sorry we didn't get a chance to meet or talk through before the session's actually started. Okay. Brilliant. How are we doing for numbers, Lydia? Should we be thinking about starting?
Yeah, we're on about 23 now, so whenever you're ready.
Okay, so why don't we do that? Okay, brilliant. Good morning, everyone, and welcome to this webinar from the BHF Data Science Center. I'm Matt Sides. I'm hosting today while Kathy's away, and I'll be giving the first presentation too, where I'll be talking to you about data-enabled trials. And then I'll be handing over to Honghan Wu, who's going to be talking to us about some of the COVID-19 work that he's been leading and involved with, which I'm very excited to hear. Now, hopefully you can hear me okay. I'm not sitting in a way that I was expecting to, so I'm slightly, I've thrown myself a little bit here, but let's see what we can do. So I'm going to talk to you about data-enabled trials and the BHF Data Science Center, and what we've ambitiously called ushering in the future here. So for those of you who don't know me, who am I? So I'm Matt Sides. 80% of the time, I'm Professor of Clinical Trials and Methodology at the MRC Clinical Trials Unit at UCL. where I've been running clinical trials for many, many years. But for the last 6-8 months, I've been seconded a day a week to the BHF Data Science Center to lead on the data-enabled trials theme. And I'm super excited to be here. Many of you I've met and interacted with, others I will not have. So if there's anything you wanted to find out about me, I'm sort of vaguely there online. I nearly gave you a link to my LinkedIn, which you're very welcome to connect to me on, but LinkedIn wouldn't send me a code to allow me to get in this morning for some reason. I've just had my laptop rebuilt and it's not working properly for me, which is interesting. So, clinical trials, what makes clinical trials different to other forms of research? And why has BHF Data Science got a theme just about clinical trials? Well, actually, clinical trials are important for us. They're the gold standard for evidence-based medicine. They're run within a complex regulatory framework, particularly if it's drug trials and we're looking for licensing, but any trials are run to a very high and slightly complicated standard. I say the important thing about clinical trials is they can directly impact on future healthcare provision. Consent is nearly always taken from participants, which is important. They're informed for what's happening and what's going through. Allocation in late phase trials is at random between 1 or 2 things that are being tested, or more if you're running a multi-arm trial. We have strict follow-up protocols, so people are being seen as and when the trial requires them. We have prospective data collection, so we're not going back and saying, using what we've got already, we're collecting new data. And we have very strict reporting standards for what comes out of the trial as well. And so it's worth bearing in mind that a phase 3 trial, those randomized controlled trials, would take hundreds or thousands of participants, can take tens or hundreds of participating sites. They may take anywhere between 3 and 10 years to get an answer, and may cost millions, tens of millions in the case of drug company trials. It costs a huge amount, and we don't want to get them wrong. Worth bearing in mind that the clinical trials are really the only route to testing treatments that are not already in use. So yes, plenty of call to use more real world evidence, and you've heard more of that on radio news this morning and places where that can be used. But for things that are new, you're not going to be able to get that out of you're not going to be able to pull out retrospectively where treatments have already been used. So why do we want to use more healthcare systems data for trials? I keep using the term healthcare systems data here, but I won't be consistent. I prefer that term to routinely collected data. If you participate in a trial, you may end up having scans or blood tests that wouldn't be part of your routine care, but there is a slot for those to be recorded in your electronic health record. So, you know, it's they haven't been collected because of routine interactions. They've been collected because of because of research. I'd love research to be routine. I'd love research to be a part of everyday practice, but that's a different ambition to today. So I think healthcare systems data is a much better term for us to use. Anyway, look, there's considerable overlap, as you would hope there would be, between the data that's needed for any clinical trial and the data that's already being collected in healthcare systems. An appropriate use of that healthcare systems data could reduce the duplication of effort. At the moment, you know, people are putting things into the hospital system, and they're also putting them separately into case report forms. Very often that's a nurse or a data manager transcribing them onto paper, sending them off to a trials unit where they get retranscribed in. That's a lot of work, a lot of effort, unnecessary duplication. It may lead to transcription errors. So we can reduce that if we take data straight out. We can then improve on other aspects of quality, you know, particularly completeness of data. It allows us, when we're monitoring, to focus on the trial data and the systems that actually need attention. And overall, this should reduce trial costs, allowing us to do more research within the limited funding budgets we have, and hopefully may even reduce the burden in patients too. And actually, there are risks in not using healthcare systems data. You may have missed events if people are admitted to many hospitals. My poor dear old aunt was admitted to 5 separate hospitals during April and May last year. I'm pretty sure if she was in a clinical trial, at least one of those would have been missed. I'm certain, just because of, you know, bouncing around systems. So have trials been accessing electronic health records, healthcare systems data? Mostly they hadn't been. We ran a review, Sarah Lenson there led the review of 81 registries. And we found of those 81 registries, healthcare systems data collators, 22 of them have provided data to 160 trials between 2013 and 2018. We estimate that's only a small proportion of trials, equivalent of about 3% of UK trials, at least compared to the number in 2015, or certainly less than 5% of trials. Most commonly, people have been going to NHS Digital, to ISD Scotland, as it was called then, to Public Health England. Obviously, there's more centralisation of where those data sets are now, of course. Commonly, it was large cancer and cardiovascular trials. Cardiovascular trials, you folk here are ahead of the game in terms of potential. And I've come to this field from growing up doing cancer trials. But what the data were being used for varied considerably from trial to trial. You can contrast this with Andrew McKay's review. He looked at NIHR-funded protocols, and 50% of those said they were intending to access electronic health records. So there's a big disconnect there, isn't there? What's different between this intention for people to access electronic health records for trials and what they're actually doing? Well, so people often talk about making our trials data enabled. It's a silly term. What trials aren't enabled by data in any form? But digitally enabled is a perfectly good term, I think. So NIHR, Health Data Research UK, and Clinical Practice Research Data Link came together to run a data-enabled trials workshop a couple of years back now, looking at efficiencies on study design, on feasibility, on recruitment, on follow-up, and on analyses. And what could be embedded? You needed to be able to embed all of those into routine data if you could. And if we did this, according to some, I think if we're embedding at least some of them into the trial, then a lot more trials can be considered digitally enabled. That paper that came out of the workshop is now available. You can read it online. And these are the recommendations that came out of here. Wider adoption, building in methodology questions, early engagement of regulators, better patient and public involvement so we can collect the data better, greater interoperability. I won't run through all of these. You can read this for yourself, but this capacity and capability building, I think, is really important here. And many of these challenges, maybe much of what that came out of that is also echoed in the survey that the BHF Data Science Centre ran last spring. 56 people responded, 35 of whom are currently or previously involved in running cardiovascular trials. And the main challenges were about gaining access to data. What were the processes? About the lack of information, the lack of guidance, about the data issues, including availability and defining outcome measures, and about the costs, not just about the costs themselves, but about the clarity around those costs. What data sets people were using varied quite a bit, whether it was primary care or secondary care, whether it was registries, death data, other data sets. And what they were planning to use as well varied, whether it was about planning or recruitment or contacting, primary trial reports and long-term follow-up, different stages at which you could be using this. Many of you will know about the NHSX find, recruit, follow approach, which is about identifying how many patients are out there, how to identify those patients, how to use them in the longer term. And they've really been emphasizing on the identification. That is important. Many trials fail to recruit, and better identification would help. And certainly for prevalent case trials, that's going to help. But for incident case trials, where the patients are coming to clinic because they're newly diagnosed with something, then that's perhaps less of an issue. And the comments here, I imagine, will resonate with many of you, and you'll have heard them. And some of those are real for sure, and some of them may be perceived. They may just be real, but it doesn't matter either way, because if it's seen as a problem, then it's a problem that needs to be addressed. So what are we doing? Well, our vision is that within 5 years, a data-enabled trial will be the default approach for every new cardiovascular trial in the UK, and that broader definition. So we want to aim to streamline the use of routinely collected healthcare data for trial planning, for recruitment, for follow-up. And it's a really interesting area because there are plenty of skeptics out there who say you can't get the data are out of date, the data are unusable. And there are plenty of true believers out there who say, well, why are you still doing trial-specific data collection? You should always be using electronic health records. And the truth at the moment is probably somewhere in between, varying on different outcome measures and different trial settings. So it's clearly an area that's ripe for guidance and ripe for research. So let me give you a very brief overview of our plan. I say we want to increase the number of cardiovascular clinical trials using routinely collected data sets. Well, we'd like to provide guidance and contribute to that guidance that's out there. So our goal is to make the necessary bureaucracy easier to navigate, make it easier to obtain ethics approval, obtain consent. We want to provide independent guidance to help trials navigate those processes. We want to make clear what datasets are available. So there will be web pages in due course on the Data Science Center website focusing on things that we think you as a community will find useful. We want to provide support packages through webinars and open clinics, and I'd really like to speak to you about whether you think that would be helpful. And we'd like to make it clearer what datasets are available. And so talk to us. A number of trials have spoken to us before they've submitted their grant applications. We're here for discussions. And there could be discussions about specific things, what datasets, how to use the data, about costs. I mean, we would like to think about going with a group of trials to negotiate for what appropriate costs would be here. So talk to us, use us as a resource here. We're keen to facilitate this. We want to develop best practice, and that involves establishing ways of accessing and using healthcare systems data to impact trials. We want to enable shareable data from trials as well that have used healthcare systems data. So one of the ways to do this is to have make a catalogue of trial case studies where people have used the data. We also want to record the timeliness with which you can get the data and say develop best practice guidelines. So the case study approach is really important. We had 2 of the HDI UK interns with us last summer who did a great job of heading off and trying to construct narrative case studies from some of the trials that accessed and used data. And those were been identified from the survey that the Data Science Center ran last year and the review that Sarah Lenson run. And as I say, we've got short case studies. We want to put those on the website. The intention is to make those available. You can then comment on how useful you find them and help us flush out more case studies, more questions about the ones that are there. We want to make these useful. We want to make you want to read these case studies. And so we need your input and your help in constructing them. And as I say, all we're doing is cataloguing. All credit to the teams that have actually been doing this, of course. And I say access case studies can be helpful. If you've had an experience, put it in the literature. I think that's really important. Arjun McNair at the MRCCTU did this, accessing cardiovascular data to be used within a couple of cancer trials where they were looking at cardiovascular outcome measures. He found a real challenge in his trying to get the data during the time he was doing his MD. And so, we mapped this out. What was the experience? We shared this with the healthcare data systems data sets. They co-authored this paper, and they're now very much aware of the timelines. But we know that CTUs need good data structures, clinical trials units, and applicants need experience as to how to answer the data set. So something for everyone. It's not just the providers of data. Applicants need to be better aware. We want to be able to use our leverage to improve processes. We want to contribute to meaningful discussions. We want to be in the room where it happens. We're knowledgeable. We'd like to be able to influence and bring our thoughts here and collate those comments, collate the input and pass on your challenges to groups. We want costs to be consistent and durable, for example. We want to be able to influence that bureaucracy. I mean, the important thing is about funding here. Trial teams really get one bite at the funding cherry. It's upfront, it's early on. But the costs of routine data are often not clear up front. Trial teams often don't even know what data they need exactly at that point or how often they want it. And if you got a quote from NHS Digital, NHS Scotland, it often lasts a very short amount of time, far less than the amount of time that's available that's taken in the grant review process. So it's very difficult to do that. Or if you've been collecting trial-specific data collection, you want to engage in SWATS, data utility studies. We'll talk about those in a second. You may want to go back and get that data, but then you don't have the money to do it. So how can we influence? How can we make that happen? Part of that community collation we ran with Gavin highlighting here in the cardiac surgery group, some workshops in October, really setting out over 2 half days, good discussion, challenges and solutions to those. And the report for that should be available on the workshop on our website quite soon. An important new piece of work is thinking about developing standard outcome definitions and code lists. We want community agreed algorithms. It's not just a code list, it's how do you connect them together? How do you, from what data sources, how connected in time and space do they need to be? What if you've got missing data? We need community agreed, say, algorithms and best practices. So we've started that. We convened an initial group. That launch meeting was last week. It's a small group, mostly clinical people. We need to widen that out. We will need you to be involved in this. It's going to be really important for us to help us define this. And say, we'll start with a few things and build out. We're calling this SCORE CVD. And I might get Jackie to put in the chat how we were defining the word SCORE, because it's a slightly lost my memory right now. And so we're doing this together with the computable phenomics theme, which Spiros is leading. As I say, we want to define what the practices may be. We want to flush out those phenotypes, those recipes for defining outcome measures that are already there. I'd say the aim is to get to better approaches. We need to identify what's been done, set out our good processes, set out our additional requirements, define with the community those defined the definitions, develop them, and then disseminate that. And the aim is to get some of those out within the next year. And then we want targeted driver activity as well. And that's really about enabling evidence that routinely collected health data can support clinical trials. And some of this is going to be about the utility of healthcare systems data, and some is some is about other experiences here. So let's pick out this. We have to co-develop SWAT protocols with you. I mentioned that, but let's first talk about there's a report that's online as of last Friday, looking at whether routinely collected health data, two data sets can be used as as source data for trials. Actually, they're effectively usable. Now we're going to be challenging other healthcare systems data holders to follow the process set out here. It's very freshly added. The paper that describes this was only submitted, hopefully this afternoon, actually. It's been led by Macy, and Macy will be with us for a webinar in May. So let's save the details on this until now, but this is a very exciting development. Okay, data utility is really important. How good are the routine data? Can we be using these to replace trial-specific data collection? Where can we and where can't we? So we did this, Sharon Love at MRCCTU and Anna Kolnowski, who was an MSc student. We got data from a gastroesophageal study, just looking at death, but they were getting death periodically from NHS Digital, and they were only seeing the patients every couple of years. And what you can see here is agreement of various data freezes. We always have more deaths from NHS Digital than we did from trial-specific data collection, but there was a few deaths that come through from trial-specific methods that the NHS Digital didn't know. It's a rather complicated graph here. I'll leave you to read this in the paper, but we could be doing more of this. Death is an easy one to do. And the SWAT that underpins this, SWAT 125, you can find it on the SWAT repository in Belfast. If you want to copy that approach on your data set, please talk to us. Very happy to run through. We need more of these. So there's some in the literature. Siam Ahmed, a PhD student working with me with my MRCT hat, has been looking at these. Been doing a systematic review for published comparisons, but there's more out there, I'm sure, that haven't yet been published. So do you have the potential to do one, or do you have an unpublished comparison? Could you publish it? Can we help you? We know of a few out there. People have presented these at conferences. They're tucked at the back of a paper and it's difficult to find. You've made that comparison. Help us if you're aware of these. Flush them out. Tell the Data Science Centre. Bring it to our attention. I say, Siam's Prospero review is listed there. One's just come out in the cardiovascular world. Charlie Harper, as part of his PhD, working with the Ascend team, really looking at a series of cardiovascular outcome measures, which is really important, showing sort of good specificity is really good. If you found it, the routine data almost certainly existed, but there's a chance sensitivity was a little bit lower here. We need more of this. One trial doesn't answer this, right? What we need is that patchwork blanket. That's why we've drawn this here. Series of outcome measures. We stitch it together. No one trial is good enough to tell us one way or the other, but each one is really important. So the work that they've done here is really lovely, and they've looked at the treatment effect as well. Great stuff and well worth a read. Let's stop for a very quick advert break for things that I think you'll find interesting. Firstly, there are a couple of jobs out with the Data Science Center at the moment. One looking, is this the right one? So data scientist, and there will be one, and the diabetes data science catalyst as well. There'll also be one coming soon to work with us on the trials theme. So if there's, if you think there's anybody that might find these roles of interest, then please point them in our direction. If you've been working with the routine data and you want to help us think about some of the methodology questions, the NIHR MRC Trials Methodology Research Partnership has a survey out called Cormorant at the moment. It's missing an R. I'll claim the naming rights on helping put together the name for this, but I won't claim the typo. That wasn't there. But Fiona Lugvigger in Cardiff is leading on this. Tell us what you think the challenges are, what the methodological questions are. TMRP has put a little bit of money in to do this, but it's feeding into HDR UK, and that's where some of this is going to go, and some of that will follow through to BHF Data Science Centre. What are the methodology questions? Not the operational issues, but the methodology here. So scan, follow the link, go through the Twitter feed for Cormorant. You'll be able to see that. And please contribute. To only take you a couple of minutes. You may or may not be aware of the International Clinical Trials and Methodology Conference. It's the largest academic conference on clinical trials methodology. It's next running, it runs every couple of years. It's next running in Harrogate in October. And if you've got SWOT comparisons that you think you might be able to look at, if you've got data utility experiences, this is a place to present them. You'll be presenting them to a community. Data scientists, statisticians, trial teams, clinicians in particular, anyone who attends this always enjoys it. There'll be about 600 or 700 people there. Very friendly, very good environment. I would strongly encourage you to be thinking about submitting abstracts. And if you can't submit an abstract, you attend anyway. And if you've got something you're not sure about, give us a shout. I'm talking here with the Data Science Centre hat on, but I'm also chairing the scientific committee of that conference. And just to mention very briefly, if you're interested in the clinical trials, the MRC Clinical Trials Unit runs now online MSc in clinical trials. And we've got a brand new statistics for clinical trials MSc that's starting up. Mostly we're thinking that's going to be for undergrads who are currently doing numerate degrees, or for people who are thinking for clinical folk who want, or data scientists who want to get an extra something under the belt. So really focused, great group going to be running that. That's the end of the advert break. Please forgive me for doing that. Just tell you very briefly about one new paper that just came out, which I think will be of interest. And I think it falls on this, one of the three hats I'm wearing here is a BHF Data Science and App. This is about guidance to protect blinded clinical trials in EHR systems. In a blinded clinical trial, at least one group doesn't know the allocation. That may be the participant, often it may be the person assessing. Very often that's done with a placebo, but it doesn't have to be, or a sham treatment. That doesn't have to be the way. We've identified, though, a couple of ways in which people could be accidentally unblinded. People may see things that they shouldn't. We kept it all very hypothetical in the short paper that's just come out in clinical trials. But in the electronic health record systems, the hospitals that are using them, there's so many role functions that actually not everybody gets protected from what they should. So in a patient portal, you might see information that tells you what you've been allocated to, for example. So this needs really careful consideration by trial teams in setting this up, and really careful monitoring by site teams. I've set out some actions here. We've also put in the appendix of this, a couple of templates that people should follow in looking at what they might do. Very helpful to discuss. If you don't want to read a 600-word paper, then there's a 200-word Twitter thread if you prefer. Let's keep our reading light, eh? Okay. So you saw our vision. Data-enabled trials, let's make it the default within 5 years. As I say, our aim is to streamline. There are challenges. We want to help work on the solutions. And we're one of many groups doing this. As I say, I'm wearing an MRC CTU hat here. TMRP is important. HDR UK is critically important. NIHR is a real driver here. And there's so many people that I could have and should have put on here, but I didn't. Anyway, I'm going to pause there, and maybe we've got time for one or two brief questions, and we'll pick up more at the end if we've got time. Anna, Katharina, you have your hand up. Do you want to turn your camera on, turn on your mic, and take your question? Welcome, Anna.
Hello. So it's a very quick question. I saw on the Oxford University website that Martin Landry is also doing something similar in terms of streamlining clinical trials. And I'm just asking how much of the work not necessarily overlaps, because I'm sure there is plenty to be done, but is it not important to build synergies? I think he wants to build on the experience of the recovery trial in terms of recruitment and working with healthcare, whilst this seems more like using data rather than necessarily building on the NHS structure of delivering the trial itself.
So Anna, the protest work that Martin has just set up is about trying to deliver what I understand is industry trials in the UK and trying to get them to use routine data. There's a lot of work to be done before industry is going to be interested in using routine data for follow-up, but they are interested in doing that for recruitment for sure. I don't know how much of this will be cardiovascular work, and as I say, it's a very new organization. I've been working with Martin on the HDI UK trials theme. He was leading that in the first quinquennium. Maria Matham from Oxford, who's been works closely with Martin on most of his trials, and I are co-leading going into the second quinquennium. The work that Macy will be speaking to you about, Martin is a co-author on that work, and Martin's very much involved in that workshop that I presented. So that synergy is there. We're trying to make sure that there's not duplication of effort, and people are very much trying to work together here. Hopefully that reassures you on that. What was the second question, Anna?
I'm a public health trainee, and I'm currently working at NICE. And there are always concerns on when people assess the evidence, where the evidence comes from, but NICE is also trying to get more real world data built into the guidelines development and how it can be accepted. And how do you see these as kind of reducing the cost of running clinical trials, reducing the downstream costs of medication for the NHS and for taxpayers?
So Anna, I think it's critically important that NICE and other commissioning bodies are seen as critical partners here. It's anybody that's going to be using the results or trying to interpret the results of a trial, we need to have in mind. And people often talk about the regulator, but to me, NICE is just as important here, if not more important, because most treatments will end up going past NICE, even if they don't need to go past a regulatory body. So you're making sure that NICE feels that the results of a clinical trial can be used and are interpretable, I think is really important. There's lots of people talking about real world data and how it can be used. And I say there will be issues, I think, where where we run a clinical trial, the kind of health services research kind of trials, where we're comparing two treatments that have already been used for some time, and the trial is really there to anchor that data. And I think real world evidence has a role to play there. If I've got a brand new treatment that's going to be looked at, real world evidence isn't going to, alone isn't going to help us, but we can use healthcare systems data to supplement, to collect that data more efficiently. I say that work that Macy's presenting is why the regulators should be happy with that data, and NICE is one of the groups that we will talk to about that in due course as well. But when the paper element is out, what I've given you today is effectively a sneak preview on that. But Anna, with your NICE hat on and wearing any of the hats I'm wearing, I'd really love to have a further and deeper conversation with you and your NICE colleagues, if we may, please. Okay. I'm very conscious that it's past already. So Honghan, I'm going to pass over to you. Would you like to share your screen and let me welcome you here? And let's make sure we've got time for a couple of questions for you as well. So Honghan, I'm going to leave the introduction to you, if I may.
Thanks, Matt.
Welcome. So we're seeing we're not seeing presentation mode. Perfect, that's great. Over to you, Honghan.
Thanks. Hi, everyone. We don't have too much time. I may have to rush some of the slides. Anyway, my name is Honghan Wu. I'm based in UCL as well. So this is a preliminary result of our CCU-019, which is looking at rare disease patients, particularly the impact of COVID on these patients. So we started this project last September or so. the motivation is simple. We would like to know the rare disease patients, the impact of COVID-19. When we started, we found very few studies in this area. The challenges could be the data and also the definition of rare disease itself actually is challenging, as you will see later. And my colleague Anna Katrina, who is CAT as for short, will have strong opinions of some of the slides I'm going to show you. So the work is done by Huayu, Johan, and Kat, and many other colleagues as well. So this is just one of the several studies we're going to do with this issue 019. Okay. So first of all, where do we get these definitions of rare disease? So we use this knowledge base called OrphaNet. I believe some of you know this. OrphaData is a data they provided from that consortium. So particularly we focus on, we follow these, some of the definitions for this paper published in European Journal of Human Genetics in terms of how to find these particularly relevant rare disease out of these 6,000 or 7,000 rare disease defined by OrphaNet. So this is the kind of pipeline. We obtain the final numbers for our analysis we're going to discuss. So we start from this whole set of rare diseases, then we select those with prevalence data, which means that OrphaNet provide epidemiology data for these diseases. This is a subset. Then for all these different types of prevalence data, we are only interested in those with point prevalence because of the studies and data set we are using in studies we are going to analyze. And then out of these 700 also, we only can do ICD-10, those with ICD-10 mapping so that we can get these patients from the TRE, do analysis, which ends up with about 700 also. Then we remove some of the disorders by removing these ICD-10 chapters. For example, we're not interested in these cancer patients. We're not interested in infectious disease or poisoning and some other ICD-10 chapters. From there, we end up with about 600. Then we want to have a very accurate mapping from OrphaData to ICD-10 mapping. So we only focus on two types of mappings provided by OrphaData. One is called exact mapping, which means that one OrphaData disease to one ICD-10 code. Then this second category is called broad term to narrow term, which means that we might lose some information by doing this mapping, but the mapping is accurate. They do have other types of categories of mapping. We think for this stage, it's good to avoid them. So that ends up with about 145 diseases. Then out of this 145, there are 11 rare diseases which we could not find any cases in our TRE. I have to say this using English data and also only use secondary care for this identification. We try to use the primary care, but it does not give us any cases of these 145 diseases at all. So these 11 diseases are not in the follow-up result set. So this is an overview about the numbers of patients with at least one of these 134, remove as 11 empty diseases. We end up with about 2.5 million patients. I break this down to ICD-10 chapters so that we have a kind of overview of the numbers of patients within each category. I highlight those in red boxes, which are kind of more relevant to CVD, this consortium, like those blood-forming organs. This is about 235,000 patients. The other is about disease of circulatory systems, which is about 327,000. So this log scale. And we are interested in to say, have a kind of quick comparison with the TRE data, English TRE data, compared to OrphaNet prevalence data. We generate this kind of two scatter plot. This left one is for those diseases which has less than 50 out of 100,000 population. on both the TRE data and also from OrphanNet. You can see the general pattern is that there are a few good alignment here and there, but there are big disparities between these two data sets. The reason could be for OrphanNet, they got this data from the systematic review. Some of these papers they reviewed have very small cohort, or some of them even though have large cohort, but it's not UK or European based. So what we did here, we pick up those mostly from this order, worldwide, European, UK, and US. But if we cannot find any of these prevalence data from OrphanNet, we just choose one of the first in the list. So this kind of missed types of prevalence from OrphanNet. And we also, plot another group, which is with less than 5 out of 100,000 population. You can see similar patterns. They do have some good alignment, but a lot of disparities over there. Okay, that's just a quick overview of the 145. For the next few slides, I'm going to show you the result of COVID-related analysis. So first of all, we are interested to say how these patients we identify from which data set. So as I mentioned already, the primary care data in the TIE could not give us any cases of these diseases. So we ignore all of them. And then most of them come from HES. One is outpatient, the other is inpatient. The first table you can see here, these are disease chapters. These are sources of the data source. These are percentage. So as you can see, for majority of these ICD-10 chapters, the inpatient care data source can give you majority case, apart from the final one, which is eye diseases. Only about, it's about 88%, but still very good, but not covering as good as other chapters. And for outpatient data source, they can provide some information, not too much, but for this particular two types of the top two ones, one's eye disease again, the other will be MSK and connective tissues, which about 12% of the patient comes from outpatient and data source. Then this slide's about the demographic information comparing general population, which is the left-hand side table, compared to other three groups of rare disease patients. The first group is all rare disease patient. The second will be the blood chapter, and the third will be circulatory system chapter. I just highlight a lot of numbers, just highlight some key interesting point. In general, these rare disease patients are older compared to general population. Over 60 is about 70, sorry. So 27% compared to 12% in general population. Particularly for these two CVD related ICD-10 chapters, they are even older, particularly for circulatory, about 58%. And for ethnic groups, and the Black is kind of high population, higher compared to the general population. And for blood chapter, Asian, British, and Black higher, much higher actually for this Black, for example, 18% compared to general population 3%. For the gender, the female is higher, but you will see later because of the disease distributions we selected. That's my partially to explain why a female is higher in this rare disease population. This is slides about infection rate of rare disease patient compared to general population. The general population is about 6%. The rare disease patient is about 10. But if we break down each single rare disease patient groups, it's about 10 as well, but IQI is about 8 to 12. In general, it's kind of much higher compared to general population. So given the time, I'm not going to talk about these details, but just skip this. If you have time you're interested in, maybe you can come back to these slides for those who are interested in this. Okay, this slide and next two slides as well is going to look at these phenotypes of these rare disease patients. So for these slides, I compare general population to three ICD-10 chapters, including circulatory systems, congenital malformations, and disease of blood and blood organ. So you can see these overall numbers over there in terms of patient had COVID-19 positive test. For circulatory, it's about 20,000, and congenital malformation will be about 17,000. These are blood and blood organs, about 24,000. And these x-axis, these different categories, which is the first one would be COVID test. The second one would be the GP confirmed diagnosis of COVID. Third one would be COVID admission. This ECMO treatment, ICU admission, MV treatment. All these are ICU kind of treatments. This is a COVID-19 inpatient death. These are fatal with COVID diagnosis. The final one was fatal without COVID diagnosis. So as you can see, the overall pattern, if you particularly for looking at this kind of severity in terms of phenotypes, the three ICD-10 chapters much higher compared to the general population. For example, for this orange bar, which is a disease of the circulatory system, as you can imagine, is higher, but it's much higher than I expected, my own kind of previous knowledge. It's about 6 times higher in terms of admission. And for other two, it's still very high, about 3 to 4 times higher. For ICU admissions as well, about 3 to 4 times higher, and similar for the treatments. And the death is about 11 times higher for this orange bar. And again, for this kind of fatal with COVID diagnosis is much, much higher. These, you will see, because the whopping green bar here. This would be compared to another 3 chapters, which is a nerve system. And second one, this green bar is respiratory system. And the other one is endocrine and nutrition stuff. So the eye-catching stuff will be in the green bar, but there's a caveat over here, as many of you can kind of interpret this. This is only one disease falls within this green category, which is ARDS, stands for acute respiratory disease syndrome. A cat looked at this slide yesterday and said, this is not right. ARDS is itself not a disease, it's just a complication of other stuff. I put this in COVID-19 scenario is kind of complications of COVID-19 caused by COVID-19. So, which means we do need to have a much better definition of rare disease, because this actually appears in alpha data and also many other rare disease ontology, if you like, define this as a rare disease. But this, of course, is problematic if you see this kind of risk of COVID. It's more like a kind of complication of COVID. Anyway, but the general pattern still holds for other chapters as well, much, much higher in all these phenotypes of rare for COVID, for this rare disease patient. So this slide breaks down one ICD-10 chapter is circulatory rare disease to each single individual diseases. This is a list of diseases. I'm not going to read them out one by one, but still you can see these overall patterns. This one is look at this, is about 8-9 times higher. These this kind of this bar and this gray bar as well, and many others. There are many numbers over there. Course of time, just skip this. But you got the idea. Rare disease patients, even though we haven't got a very good definition of rare disease, but the overall pattern is very clear. They are very severe, much more severe compared to general population. We do need some kind of public health policy to be adapted for these patients. That's clear message we can send out. Okay, next is about some multivariable analysis. Huai, do you mind to introduce this briefly?
For the sake of time, I will only do this briefly. So this is as a multivariable analysis from the cohort developed by CCO 13, and basically these are positive tested population, and in the analysis we included 3 diseases, so the hereditary ataxia, HHT, and Marfan syndrome, because we identified in a smaller cohort previously in Genomics England that HHT and hereditary ataxia had a higher risk of a severe COVID-19 outcome. And then with Marfan syndrome, we didn't see a higher risk in the smaller cohort. So we want to confirm this in a preliminary multivariable analysis in the CVD COVID UK data. And so the conclusion from this analysis is that indeed hereditary ataxia and HHT, they have independent contribution to the COVID-related death, with the odds ratio and the range you can see in the text, while it is not the case for Marfan syndrome. And I think before there's no clear, solid evidence-based claim that these patients are at a higher risk for COVID-19, but this is with this consortium, this is what we the evidences we can deliver now, which was not possible in other data sources or in other or previously. Yeah.
Thanks, Raj. That's brilliant. Okay. Just a few words about next steps of this CCU 019. So first of all, we need, as we already mentioned, we need a better rare disease definition, and Kat is very nicely offer her time to go through all these rare disease we extracted from offer data. She's going to review, she started already actually, review them one by one, just make sure these mappings, these definitions put in the context of our study makes clinical sense. That's very, very important. And also we might think of kind of time dependencies in this COVID-related analysis. For example, whether this diagnosis is before or in the middle of the COVID-19 pandemic, that might give us better way to interpret that data. That's first thing. The second would be focus on this kind of outputs. As the first outputs, one would be the one I'm talking about. The other would be potentially look at this rare disease epidemiology among all these 57 million people compared to offer data, the paper I showed previously at the first slide. Then the second output of this work, we haven't started. We started a little bit, but we haven't full speed on this. Is kind of using machine learning to identify those patients who might not, officially diagnosed as rare disease patient, but based on some machine learning model, they might share similar kind of vulnerability like those who had a clear diagnosis, so that we don't miss those high risk patient groups. That's the second focus of the CCU 019 study. That's everything from us. Matt?
Thanks. Thank you, Honghan. That's great. Can I just share again